So what we're going over today is the Calvin cycle, the second part of photosynthesis. And there are, of course, two parts that we have been talking about, the light-dependent reactions, okay, which happen inside these little discs or cookies that we call thylakoids. And we're going to now talk about the light-independent reactions. Kind of misleading because both reactions require light because we need to have light to produce ATP that is needed to create the G3P, which is a compound that leads to glucose in the Calvin cycle. So this does not really occur when there is no light. So sometimes I call these the dark reactions. So they're all driven as a non-spontaneous process by the light from the sun that these photosystems pick up. Okay, so we've already handled the light-dependent reactions. So we're set up for the Calvin cycle, named after Dr. Melvin Calvin, who won a Nobel Prize in 1950 for understanding all of the reactions that occur. So to set this up, we should understand that the light-dependent reactions produced ATP that is necessary. All the ATP that was produced by the proton motor force that was created by the electron transport system here, and having these H pluses or these protons move the turbine of the ATP synthase in a way that will force the ADP to slam into phosphates to make ATP. All of that ATP is going to be used in the Calvin or the Melvin Calvin cycle or Dr. Melvin Calvin cycle. Too much to say, we'll just say Calvin cycle here. All right, that's really important. You understand that the entire amount of the light reactions were to set up the Calvin cycle. Not only are we taking all of the ATP to anabolically create glucose, or in this case G3P, which is a precursor, it's a 3-carbon precursor to glucose, which we know is 6 carbons, and other organic molecules the plant may need. So besides the ATP that we're creating inside the thylakoid, we're taking the high energy electrons that were stripped off the water. Okay, we split the water, remember the light reactions, and these electrons traveled through the chains, and eventually they were re-energized after they were used to pump the protons in, and they're now being absorbed by the NADPH. We should recognize the fact that one H2O delivered 2 electrons through this process. So it's understandable that we should understand how many ADPHs are being produced in this process, and we'll go over that. Understand all of those NADPHs, which again are just a high energy electron carrier, is delivering 2 electrons and a proton. Okay, and that's all being delivered into the or setting up the Calvin cycle. And that, of course, is occurring in the stroma outside the thylakoid. Okay, very important we understand where the reaction occurs and what are the reactions. So understand that ATP is coming through, and also Na what DPH, okay, is being delivered into the stroma through this system. Without those two, there are no light reactions. Okay, so let's get a bit of an overview of these reactions. And it gets a little bit complicated. I'm going to take you through a little bit different way to explain this, okay? So the first thing you understand, there's 3 parts to the Calvin cycle. There's the carbon fixation. And now all that means is the CO2 in the air, and it gets into the leaf by these stomata, those little openings that we'll talk about, or you may have already talked about this in your class, but the CO2 needs to be delivered to an enzyme that we call ribisco for short, and that's nothing more than ribulase biphosphate carboxylase, ending in ase here, to make some sense of all of this. So it's an amazing process that we're going to take CO2 from the air and we're going to combine it with a RUBP, or ribulose biphosphate, okay, compound here that's a 5 carbon. If you notice, there's 5 carbons here, and these 5 carbons are going to combine with this carbon to make a 6 carbon molecule, and that's going to break apart into 2,3 PGAs, and through some ATP. This is going to run twice, okay, because we're going to have now 2 3 carbon fragments. Remember, a 5 carbon fragment is going to gain a CO2 and become a 6 carbon fragment. And by the way, the carbon that we're adding from this CO2, I'm making in blue here, these, or the 2 carbon fragments, going to break apart. And then they're going to run through twice here. And we're going to add more phosphates. And then we're going to deliver the high energy electrons and a very important proton. Okay, and I say that here because we're going to deliver 2 high energy electrons, and then we're also going to deliver an H+, and I'm going to show you where that H+ is. And you say, well, why is that important? I don't see that in other videos, because the electrons in a carbon to hydrogen bond are the high energy electrons that actually get harvested in cell respiration. Okay, so we're creating the high energy electrons non-spontaneously. Delta G is definitely going to be positive here. It's going to be forced to happen, okay, because of the light that's driving this or coupling this reaction. And we're coupling these reactions with ATP, as you can see in different places. Okay, so we have the carbon fixation. We're going to add the carbon, and we're going to basically have a 6 carbon, when you put these together here, a 6 carbon molecule that gets split. And then we're going to reduce these 3 carbon fragments now by essentially giving them electrons. Remember, reduction is gaining electrons. And so we're going to give them 2 electrons, each to these 3 carbon phosphates, and make something called a G3P. And that G3P, at least one of them, okay, is going to lead to something called gluconeogenesis. We're going to make glucose from this. Notice it's a 3 carbon, okay, fragment that gets made in the reduction phase. Reduction is gaining electrons. And that is going to combine with other G3Ps and eventually make glucose, okay? Now, half of these 3 carbons or G3Ps we make have to be put back into the system in the regeneration phase to regenerate, okay, the 5 carbon molecule RBP that needs to run the cycle again. Remember, this is a cycle. This is not a linear process where we're going to absorb carbons from the air and keep adding them to them. We're going to put them into one spot, and that one spot is going to be recycled into the next spot and be reused. So what's really important to understand this carbon or this Calvin cycle is the cycle is preloaded already. And that's really hard to understand initially, because you think about, we're going to add a carbon, then add another carbon, and that's just not the case. We're going to add a carbon into the same position every time, and the cycle needs to repeat itself. And eventually we make enough, or we add enough carbons to the system to make glucose, which would be 6 carbons, and we'll talk about that. But the biggest part of reduction, of course, is making these G3Ps that will lead to glucose. Half of them have to be regenerated into the RUBP. And if you notice a 3 carbon fragment, how does that become a 5 carbon fragment? And we'll get to those details at some point. So let's just jump into the first cycle. Let's look at the details, or the first part of the Calvin cycle, called carbon fixation. So looking at the first part of the Calvin cycle, carbon fixation here. And as I said before, CO2 is going to combine with a preloaded RUBP molecule. And here it is. It's called ribulose 1,5-biphosphate. We'll just call it RUBP for short. And you can see the carbons. Carbon makes 4 bonds. And I've got this model here where we got the phosphates at the end, noticing that a phosphate, of course, is going to be bonded, okay, to an O. And let's just draw that in here for fun. Okay, so we're going to have a phosphate, and it's going to be bonded to an O, and it double bonds to another O, and it's got these other single bonded oxygens that are usually negative, or they can have an H there. So this is going to bond to that carbon. So when I draw that phosphate there, that's what I'm talking about. Okay, so back on this, back to reality here, let's continue on. All right, so we're going to have these phosphates. You can see them here and here, and I have them in the molecular diagrams. And it's very important to understand that this is a preloaded system, meaning it's a cyclic compound. As it turns, we make more of this assembly line type of system that's cyclic. And we'll keep talking about how important that is. In any case, we're going to start with ribulose 1,5-biphosphate, and it's a 5-carbon system, as you can see. And it's going to enter something called Ribisco, which is short for ribulose biphosphate carboxylase. And it's probably the most important enzyme on the planet, maybe the universe, again, um, in terms of living things. Why is it so important? Because, well, all life really begins with producers. Producers are organisms that are able to generate organic compounds that other molecules can use for energy. Cell respiration basically uses sugar, glucose, as an energy fuel. We're making an energy fuel here. So this Ribisco, okay, helps us basically fix the CO2 out of the air. It's a very complicated enzyme with a structure that allows CO2 to be captured and put into close proximity to an RUBP. And so this enters the very important enzyme, and the enzyme allows for an area, okay, in its active site, if it's an active site, okay, to have something called isomerization, meaning that it allows for this molecule to move electrons around. As my arrows show, this is a pair of electrons moving to this bond here, and this bond pushes out. And it's not important you understand this, but the bottom line is this pair of electrons will jump to the electron to be negative. Okay, this will be a negative charge. This electron jumps here and leaves this to be a proton, which will move over here, and that's what I have here. So I kind of slipped it around. So this H is going to be lost as an H plus, and then it's going to find this oxygen. So this pair of electrons will jump here, and these electrons will be forced to jump here. And this will make this negative, and the H will move over, and that's what I have here. Notice I have this double bond here. So the Ribisco creates an environment for this isomerization. Isomerization just basically is just kind of reshuffling where atoms are. It's making an isomer. It's a structural isomer. The number of carbons and hydrogens and oxygens are the same, but they just kind of moved around. More importantly, I've got a double bond here. And if you go into a little high level chemistry, not that high level, this double bond here is a pi bond. And these electrons, okay, are going to be really attracted to this positive area of this carbon because oxygen pulls these electrons away. So it's kind of like a nucleophilic attack here. But so these electrons, remember, CO2 has to be in close proximity to this area. So Ribisco creates the active site and the environment for the isomerization to occur, and more importantly, for the CO2 to be in close proximity. Okay, and so essentially this bond of a pair of electrons attacks this carbon, and it makes this bond here. Now, of course, it will push out a bond because carbon needs to have a stable 4 bonds. So this will be negative, okay? And in the process, when CO2 bonds, we'll have this this carboxylic or COOH group here at the end. So that's what's going to happen. So I'm showing you that this carbon, which is in blue, is the carbon that came in through the stomata of the leaf, okay, got to the Ribisco inside, of course, the stroma of the chloroplast, okay, in a plant cell, okay, for this to happen. But we're going to basically monitor where this carbon was added, and I have this diagram here. So in any case, we've got this added. And by the way, by this pair of electrons being kind of donated to this carbon or attacking this carbon, this pair being pushed off and now being negative, this H comes off, okay, and then, of course, that H plus is going to reappear probably on this negative here. And I kind of, redrew this over. So this will be the negative one that's here and so forth. Bottom line is, for those not following through the mechanism, that we're adding a carbon, and I'm just showing you where that carbon is now being added to. Okay, and I'm showing you that here is the blue carbon. So now we've just added, we had a 5-carbon RUBP, and now we have now a 6 carbon, an unstable 6 carbon, okay? And it's got a name, it's got a fancy name, 3-carboxy keto aberranitol 1,5-biphosphate, but it's not going to last very long, okay? With water, let me call that a condensation reaction, it's going to grab water, and this water is going to attack this carbon, and essentially it's going to break apart this molecule into 2 3-carbon species, okay? And they're essentially the same when you break them apart. So the water attacks this carbon, which is actually negative, electrophilic attack, I think in this case, okay? And it pushes this out to be a negative. And of course, some reshuffling of the protons around. This bond will go, that was once in the water, jumps to this position here. So that's, we got a double bond here. It's going to push this bond and break this fragment apart. Okay, and that's the mechanism. And then we get 2 of these 3-PGA molecules. Okay. And of course, I should probably show them a little better. And that's what we have here. So important that a 5-carbon RUBP, okay, that's already preloaded in the cycle, okay, connects with a CO2. It makes a 6-carbon, okay, fragment, which we say, oh, it should be glucose already, but it's not. And I don't show that here because it quickly, with water, notice I have the water coming in here, okay, is going to break apart into 2 fragments we call 3-PGA, okay? All right, so it's, again, 3-PGA is a short name for 3-phosphoglycerate, okay? And now we have our 3 carbon fragments. We have 2 of them, okay? And we're done with carbon fixation. Notice we're keeping track of where that carbon came on in this system. And that's again, the end of fixation here, all right? And again, a lot going on here in terms of the mechanism. It's not important you understand the mechanism. For those that are at a higher level biochem course, hopefully that makes some sense to you. We have some very important, the most important enzyme here is called the Rubisco that runs this, okay? And most of these reactions occur in there. All right, so let's go on to reduction. So now that we're going to start with 3-PGA, and now as a reduction means we're going to have to give it its high energy electrons. Don't forget, the most important piece to all of this is that we're trying to build a high energy compound from a low energy compound. And we started this process in the light reactions. Don't forget, we started with water. Water has very tightly held electrons because of that oxygen, okay, with the electrons with the hydrogen. And we took those electrons off and we gave them energy. We used some of it to pump the protons. And now we re-energized it with photosystem 1, and NADPH is now going to deliver those high energy electrons and put them into and charge this molecule. So let's get into the reduction phase. All right, so now we get the 3-PGA, okay, or the 3-phosphoglycerates, and we have 2 of them. Don't forget, we produced 2. We had a 6-carbon unstable molecule. We started with the RUPP that was preloaded with 5 carbons, added a CO2, and now we have the 6 carbons breaking apart into 3. Now we've got them, and now these are the same reactions. Just understand that in the cycle, we're running this 2 times. So we're going from this cycle here is running 2 times because we have 2 3-carbon fragments. So if you notice in my cycle here, I have ADP going to ADP, but that's going to happen twice. So I'm just showing this right here twice, okay? And again, we had Rubisco as the most important enzyme in carbon fixation, but now we've got 2 others. We had phosphoglycerate kinase, notice the ACEs, and then we've had glyceraldehyde 3-phosphate dehydrogenase is the second part. And so this is all part of reduction. The first part is we're going to take a phosphate from ATP and we're going to add it to one of the carbons at the end here. So if you notice, that's essentially what we're doing here. And this kind of energizes this substrate by adding the phosphate. And it allows for the acceptance of this electron here. So the first step, again, they're the same, whatever fragment you're using. I'm showing that this molecule was the molecule that actually accepted the carbon in the carbon fixation, as we're going to follow its flow through the cycle. Okay, so we're going to add ATP. We're going to basically add a phosphate to the end of this, as you can see. They're just kind of rearranged differently. We have the COOH ending on this side and the COOH ending on this side, and we're going to add the phosphates to them here. All right, and then the second step, probably the most important, is we're going to deliver 2 electrons and a proton, okay, to a certain position using this enzyme here. And so in the process, a phosphate's going to be removed and replaced with the 2 electrons and the H+. And that's what I have here. These 2 electrons that were added are the electrons that the carbon and hydrogen are going to bond together, okay, in these positions here. So notice we took off the phosphates, replaced them with a carbon-hydrogen bond. And it's important that you understand that this carbon-hydrogen bond is the high energy bond that we harvest in cell respiration, and that's what we're building. Now you may say, well, what about these other carbon-hydrogen bonds? How did they get there? Well, they got there from previous turns in the Calvin cycle. Don't forget that this is a preloaded system that has turned many times over before we've actually gotten to our turn with this single CO2. I'm going through a single turn, and I'll show you toward the end of this video what it means to be going through and building an actual glucose and a G3P. All right, so any case, so we've built now this G3P at the end of the reduction phase. We have 2 G3Ps that have been made. All right. And these are the precursors for glucose. They're a 3-carbon molecule that will undergo gluconeogenesis to make glucose. We know glucose has 6 carbons. This has 3 carbons, so we should recognize that we need 2 G3Ps to do that. But understand that in our single turn with a preloaded RUBP, okay, you should understand that one of these G3Ps that we made at the end of reduction by, again, forcing 2 electrons in this area here and adding the proton and making this high energy carbon hydrogen bond here, one of them is going to go to make glucose. All right. The other one is going to be used to regenerate, okay, the starting, compound that's going to essentially be able to absorb another carbon and keep the cycle going. All right. Now it's kind of interesting that we only have 1 carbon in our 1 turn that was added to 2 carbons that were already there. Well, how'd they get there? Well, they got there from previous turns of the Calvin cycle. That's so important that you understand that. All right, that this thing has turned multiple times over. And so now the other G3P we made, although it could be used to make glucose as well, it has the same exact arrangement, again, has to undergo the third part, regeneration. We have to somehow take this and regenerate it back into the starting position so we can keep the cycle going. Remember, we're just talking about 1 turn. So you can see that 1 turn, we only produced 1 carbon that was from 2 previous carbons that were already there. So we really haven't built anything from 1 carbon. We would need more turns, okay? And I'm going to get to that, but I want to talk about how in the world, remember, we produced, let's go over this again. We had a 5 carbon, accept a carbon from CO2 using Rubisco. It made a 6 carbon. It split into 2 3-PGAs, which we started at this slide here, the presentation, and it split into, and basically it goes twice, and we made 2 3-GPPs, and understand this goes twice, and also we have to understand that there was 2 NADPHs used too, as hopefully I made that clear, okay? And now we have 2 3-G3Ps that we can use, glyceride 3-phosphates, which are the precursors for glucose. Only one is going to go to make glucose. The other one is somehow going to regenerate, for regeneration, make their RUP. But here's the problem. There's 3 carbons in the one G3P, and this has got 5. Where do we get the 2 other carbons? Okay, so let's go to the regeneration part of this and talk how that actually occurs. So regeneration, the third phase of the Calvin cycle, is sometimes called the carbon or the carbohydrate shuffle. Okay, this is not my work here. I've given credit. So this is the shuffle, how we take G3Ps and through this metabolic pathway, we create Ribisco. As I said before, in one turn of the cycle, we made 2 3-GPs, 1 carbon was added, 1's being used to make glucose, and 1 is going to be used to somehow make more of the RUBP, which is problematic. And also notice we're going to need some ATP in the process. How in the world does that occur? How can we take something that has 3 carbons and make 5? Well, we don't. We don't create carbons out of thin air. So understand, I said before, the problem with understanding the Calvin cycle is to understand it's not linear. What I mean by that is we're not taking the carbons and then building other carbons and building other carbons to make our glucose or our G3P. It does not work that way, okay? Because if we do it that way, the second carbon we add will be in a different position than the third and the fourth, and that would just be incredibly difficult to do. We're always going to add the carbon in the same position, and then we'll grow it through the cycle that keeps going in the same pathway. Incredibly efficient way to do it, but it becomes a little more challenging to explain because the cycle isn't by itself. It's already preloaded by running a few times. So if this was the first turn of the cycle that I just explained to you, there's no way that 3 carbons can become 2. As I mentioned, this G3P that was made, let's pretend that this one is going to go to make glucose. We had 2 other carbons here that we did not see add. That came from 2 other turns. This carbon and this carbon was added to the system by 2 other turns. So that would mean to truthfully make 1 G3P, we would need 3 turns of this cycle. That is very, very important. Okay, very important. It doesn't work linear, like we add 1 carbon and we're going to be done. No, this has to already have turned 2 times for the cycle that I'm talking. To eject 1 G3P, this must have already turned, like I said, multiple times because this is already loaded. So let's think about this for a process. So let's think about this running 3 times. Okay, and if it runs 3 times, we're going to end up with 6 G3Ps. I hope you can get that. Good. Now, because we add the CO2 to the only 1 carbon gets to a 2 carbon system, you can see that the 3 turns gives us the 3 carbons on 3 different G3Ps. People will explain this and say, no, 3 turns on the Calvin cycle makes a G3P that you just added. Theoretically it does, but it really doesn't, right? We never had these 3 carbons in 3 turns add directly, okay? So it's important you understand that. We talk about theoretically 3 turns is going to make, okay, a complete G3P, but those 3 carbons are on 3 different ones. Okay, any case, because this is preloaded, okay, and we added a carbon. Let's say this carbon I added, is this going to be this G from this carbon? Understand we make 2, correct? So let me just put my little drawer here. So this one we produced in our single turn. We also produced this one from carbons we have previously what? Already added. Now, because there was 2 previous turns, if you want to think that way, well then we produced what? 4 more of these. In 1 turn we produce 2. In what? 2 more turns we're going to produce 4. That should make sense. We have a total of 6. Now this is important, okay? This part is extremely important to understand. All right, so this one here is going to go to gluconeogenesis that we talked about. It's going to make glucose. These 5, including the 1 that we just made, so the one we just made plus the 5, okay, or I should say the 4, right? Because we made 2. There was already 2 turns, okay? Let's pretend 2 turns already have occurred, all right? So we produced these 6. Okay, this one's going to go to what? Gluconeogenesis, okay, to make glucose. So we're left with what? 5. 5 what? 5 3-carbon molecules. So we have 5 3-carbon molecules. And if you 5 times 3 is going to give you 15 carbons. And through this series of crazy amount of reactions and a bunch of enzymes, the ACEs here, in this crazy metabolical, it's pretty metabolical process, where we have the regeneration of, we take their 5, look at this, the 5 G3Ps, and if you want to follow this, which has 15 carbons through this crazy metabolical process, okay, using a bunch of enzymes and all kinds of pathways. That's why they call it the shuffle. It's a crazy amount of movement going around here if you want to follow all of this. We're going to make, at the end of this, 3 RUBPs, which are 5 apiece. 3 times 5 is 15 carbons here. And we started out with 15 carbons here. We started out with 5 of these individuals, or these molecules. They're personal to me. Okay, and we had 15 of them, and now we are left with 15 at the end. But the RUBP, through this crazy amount of metabolic pathway, is going to make, all right, three 5-carbon molecules that I have written here. So we have maintained the 15 carbons and we've reshuffled them into 3 RUBPs, one of which can accept another carbon and re-go. So the idea, very simply, is that we do this as a single turn. And truthfully, we need 3 turns to actually produce enough carbons here to theoretically produce 1 G3P. Even though we're not making one, these carbons are going to be just 1 carbon of each 3-carbon molecule. We need 3 turns to take in 3 CO2s to put 3 carbons into the system that are building a G3P. Okay, and in that process, those 3 turns will make 3 RUBPs, one of which can now pick up a CO2. Does that mean this cycle has to go 3 times and stop? No, it's preloaded. The first 2 turns are already there. Okay, it's important you recognize that in 1 turn, we used up 1 RUBP, but there's still what? 2 more to keep going for the next turn. Okay, and as we add one of these G3Ps is added to the system, there's already 4 of them, sorry, there's already 4 of them waiting, and the cycle continues. Okay, so it's important that you understand that regeneration is nothing more than a complex set of reactions where some of the G3Ps will come together to reproduce the ribulose 1,5-biphosphate. I'm not going to test you on how we do that, and again, some courses could do that, but understand it's a metabolical set of reactions. Okay, let's go on and look at the overview now. All right, so let's look at the Calvin cycle one more time and take a look here. If we were to think about 3 turns, again, the idea that it only takes 1 CO2 per turn, but we're using the analogy of 3 turns to understand how we regenerate the RUBP. Okay, so 3 CO2s come in. there's going to be 3 water molecules. So we're going to take in some water, and then we're going to make, what, 3 6-carbon molecules get split into 2 of these 3 PGAs. Each PGA is going to take 1 ATP, but now we have 6 if we're combining, 1 turn into 3 to make sense of this. And of course, we're going to make this intermediate 1,3-BPG, which is basically now going, the reduction phase is going to gain, and I should have 6 here, okay? So there's going to be 6 NADPHs to make this as we deliver 6 high energy electrons and an H here. So this definitely is a 6 here. And more importantly, we're adding also the H. So notice the hydrogens that are bonding to the carbon with those high energy electrons right there. Now, as I mentioned, if we think of 3 turns, we have enough for 1 G3P, but understand 1 turn will eject 1 to make glucose. Remember, glucose is C6H12O6, so we're going to need 6 complete turns to make glucose. But let's just talk about G3P. So 3 turns, okay? Every turn it ejects 1, but we're thinking about 3 turns to show how the regeneration. So that's what they always do when they explain this cycle. They talk about 3 turns at once. We should recognize the fact that this is a preloaded system. You don't need to do that, but again, it's easy to think about 3 turns will make 6 G3Ps from the 6 of these intermediates, and we're going to always use 1, okay, to make our glucose here. The rest of these 5 that are preloaded are going to recombine to make, remember, 3 times what? 5 is 15, and they're going to go through that metabolical carbohydrate shuffle to make 3 5-carbons and still give you the 15 total. And we can start the process. So this is a way that people do it. People get confused though. It doesn't go 3 at once. So if you think about it, 3 CO2s, 3 H2Os, 6 of these, and also 3 ATPs go to ATP as we do that. And again, if we just had 1 single turn, we're just making 2 of these, 2 of these. We're going to make 2 of these. 1 is ejected and goes to gluconeogenesis. The other one is going to combine with the 4 other ones that are already in the regeneration, okay, waiting for it because it's preloaded, and it's going to make 3 more. So hopefully that gives you a better overview. All right, so let's talk about an overview of what's happening between the light dependent reactions in the Calvin cycle here. Remember, we needed definitely 6 NADPHs, okay, if we're going to theoretically make and add and fix enough carbons here to make 1 G3P, right? We have to accept 3 carbons in this cyclic system to produce enough carbons to theoretically make a 3-carbon molecule. It's a precursor. Let's think about that for a second. So we have 6 NADPHs, okay, 6 NADPHs that are needed, and each of these hold 2 electrons. Okay, where do these electrons come from? They came from splitting water in the light cycle. And we know that each water molecule is giving off 2 electrons to the P680 pair of chlorophyll molecules. Okay, so we would need a total of 6 water molecules to run through the light dependent reactions that eventually will be collected by the NADPH and delivered into the stroma for the Calvin cycle. Okay, now how much ATP? Well, based on what we've done here by a theoretical 3 turns, we have 6 ATP with 3 more. Okay, so we need 9 ATP that have to be generated from the light dependent reactions from the thylakoid that are driving this process. Remember, we are basically using the energy of the reducing power of the electrons and the power of ATP to build something. We're adding carbons. This is an anabolic, non-spontaneous process. But all of this makes 1G3P. So what do you think is necessary to build a glucose? Well, we would need 6 turns, okay? Because in that case, if you've got 6 turns, okay, you're going to produce an added 6 carbons to the systems. And they get reshuffled to different places, okay, in the different molecules eventually, but eventually we're going to kick out, every turn we kick out 1G3P. So we would need to certainly have 6 turns of the cycle, and understanding there would be 6 CO2s, 6 water molecules. Oh, we're using 6 water molecules. Now hold on, we would need how many ATP? Oh, well, we would need 12 here. We would need 6 here. Okay, so you could see how all that changes for glucose. And we would need a lot more NADPH, wouldn't we? So for building a glucose molecule, these number become 12 water molecules are needed to make 24 electrons. And this would be 18 to make a glucose. This would be 6 turns, okay, here. And you should be able to understand how many of each are needed to do all of that. All right, so that's the big picture understanding of what's going on here in the stroma. And you know, this G3P that's undergoing glyconeogenesis doesn't always have to make glucose. It can make other things like fructose. It can also make all different types of other organic molecules that and I say organic, carbon base, that the plant needs. So that gives you a way to think about the entire process here of photosynthesis, and especially the Calvin cycle. Some details you need, some you don't, but I hope that helped in a lot of ways.